Hello, hi, a very happy good evening. So this is Krishna Ma'am. We've been talking about the story of life on earth, and that's biology. So thank you so much for joining my session this evening. So here we are talking about biomolecules lecture 5. So we've been talking about a lot about biomolecules. So we've been talking about the structure of biomicromolecules, or simply biomolecules, or the structure of biomacromolecules, where we spoke about the structure of protein, your carbohydrates, and your nucleic acid. So today we'll go a bit in deep and talk about the nature of bond linking once again. So I will explain you with a picture. Then we'll talk about the structure of DNA, that is your nucleotide, or your nucleic acid, the polymer of nucleotide or the nucleic acid. Then we move into what is called as metabolism, which happens in your body and my body. So with that we'll pause, and then the last lecture we'll talk about enzymes. So with that we'll come to a grand end of your biomolecules, after which we'll have a quiz based on this topic. All right, yes, ready? So here we go. So biomolecules, this is part 5. So this is lecture 5. So what have we been talking? So we have been talking about the structure of biomicromolecules and biomacromolecules. All right, so let's move further. So we had two pools, right? One was acid soluble pool, one was acid insoluble pool. So acid soluble pool is called as our filtrate. This is our retentate, right? So this filtrate has biomolecules or biomicromolecules. So this is biomacromolecules. Okay. So this, under this you have your amino acids, then you have your nucleotides, so then we have the lipids, right? So under the biomacromolecules, so we have your polysaccharides, we have proteins, so then we have nucleic acids, right? But there's a twist to the story. Lipids actually belong to biomicromolecules, but it comes in this fraction because it is always conjugative. So much we know so far. So we have lastly been talking about the structure of protein and the structure of polysaccharide. To give you a quick recap of the protein and the polysaccharide, so here we go. So talking about the structure of protein. So proteins are usually heteropolymers. So before that I can say they are polymers of amino acids, right? So they are mostly heteropolymers. So they have different amino acids arranged in a linear sequence, right? So when I write a peptide chain, I write like this: amino acid, second amino acid, third amino acid, and fourth amino acid, right? So I keep on adding the amino acid to my right end and not to the left end. So this end is called as the N-terminal end, and this is my C-terminal end, right? So writing the structure of an amino acid, so this is H, so this is your carboxylic acid, so this is your R group, and this is your amino group. So this side it is the N group, and this side it is the C group. So we have 4 structures of your proteins. So we have the primary structure, secondary structure, tertiary structure, and the quaternary structure. So the primary structure is a linear chain of amino acids. Right? So it just gives you the information on, about the order in which the amino acids are arranged, right? So you have the secondary structure. So secondary structure is made up of alpha helix and it is attached by beta pleats. All right. So you have tertiary structure. So tertiary structure is what makes up your subunits. So your quaternary structure takes up 2 or more subunits. Like example, we spoke about hemoglobin. So where you have 4 subunits, so you have 2 alpha subunits and you have 2 beta subunits. Right? So this is how your structure of proteins are. And we also spoke about how 2 amino acids are linked together in a polypeptide chain. So there's a bond formed between the carboxylic group of one amino acid and between the amino group of the next amino acid. So the bond that is formed, it is formed by the elimination of your water molecule. So you have CONH. So the water molecule is getting eliminated and you have the rest of the structure as it is. So this bond is called as your peptide bond. Right? So this is what we were discussing in the previous class. So this is the structure of your proteins. So now let's move forward and talk further about your polysaccharides, because in the previous class we were also discussing about polysaccharides. So talking about polysaccharides. So saccharides means sugars. So poly means many, right? So they are polymers of monosaccharides. Right? So monosaccharides are individual units of sugar. For example, so these can be both homopolymers as well as heteropolymers. So they can be both. It is not necessary that they have to be heteropolymers or homopolymers only. So one example that we can take is we can take a cellulose. So cellulose is a polysaccharide. So it is made up of repeating structure of glucose. So glucose is a monosaccharide and cellulose is made up of many linear arrangements of glucose, right? So this is a polysaccharide now. Fine. So then we spoke about starch. So starch is the storage form in your plant. So this starch has helical structure like this, and in between the helix you have your iodine store. So that is why to test the presence of starch you use iodine, right? So starch is also a polysaccharide. So then we also read about inulin. So inulin is also a polymer of fructose, if you remember. So when I wrote this polysaccharide chain, you saw me only adding glucose molecules to the right hand side and not to the left hand side again. So thus only the right hand side can accommodate the incoming monosaccharide. So the right hand side or the right hand end is called as the reducing end. And this is called as the non-reducing end. Okay, yes. So after this, if you remember, we went on to talk about modified polysaccharides. Yes. So where you have your cellulosic fibers. So your paper pulp, these are all your modified polysaccharide. So you have your chitinous exoskeleton. So what is so special? They have amino modified sugars, like they have N-acetyl galactosamine or glucosamine. So these molecules are present. So that is why this is a modified polysaccharide. And the bond between two monosaccharides in this arrangement, this bond is called as the glycosidic bond. So it is again formed by the elimination of water molecule. So these were the concepts that we were discussing in our lecture on Monday. So I am continuing now from the new topic. Right? So nature of bond linking. So in a polypeptide or in a protein, so amino acids are linked by peptide bonds. Yes, so I showed you how a peptide bond is formed. So peptide bond is when the carboxyl group of one amino acid, it reacts with the amino group of the next amino acid and it causes the elimination of water moiety, that is dehydration. So similarly, this is in a polysaccharide. So PS here is your polysaccharide. So individual monosaccharides are linked by the glycosidic bond and they are formed by dehydration. So glycosidic bond is formed between the two carbon atoms of two adjacent monosaccharides. Right? So in a nucleic acid, phosphate moiety links 3' carbon. So this is in a nucleic acid, so you have the phosphodiester bond, right? So 3' carbon of one sugar of one nucleotide to the 5' carbon of the sugar of the succeeding nucleotide. So in the 3' end we have the OH and in the 5' end we have the phosphate, and that is where the linkage happens. Right? So the bond between the phosphate and the hydroxyl group of the sugar is the ester bond, so which in turn forms the phosphodiester bond. So this is the peptide bond. I have shown you this when I was giving you a recap. So here you have the glycosidic bond. So this is the first carbon. Okay, so probably we can say 1, 2, 3, 4, 5, 6. Yeah. So first carbon, second carbon, third carbon, fourth carbon, fifth carbon and sixth carbon, right? So here again, so first carbon, second carbon, third carbon, fourth carbon, fifth carbon, sixth carbon. So the link is between 1 and 4th carbon. So this link is your glycosidic linkage between two adjacent monosaccharides, and this is between your nucleotides. So I have my sugar here, I have the nitrogenous base here. So first carbon, second carbon, third carbon, so you can say fourth carbon and the fifth carbon, okay. So the fifth carbon, so the 5C, so the 5C has your phosphate group, right? So it has your phosphate group, and here it attaches with the 3C. So here we read, right? So between the 3C of one sugar and the 5C of the another preceding sugar. So 3C and 5C, so this is your phosphodiester bond. Alright, so nucleotides exhibit a variety of secondary structures like your DNA, so it is your double helical structure. So here in your DNA, so having done your molecular basis of inheritance, we know the two strands of DNA are antiparallel, right? So they have a sugar phosphate backbone, and the sugar and the phosphate, it forms the backbone, and the nitrogen base are perpendicular to them, right? Yeah. So always A bonds with T with the help of 2 hydrogen bonds, and G and C are bonded together with the help of 3 hydrogen bonds. So each strand has a turn of 36 degrees and one full turn has 10 base pairs here. So the height is 34 Armstrong and the distance between two nucleotides is 0.34 nanometer. Okay. So here we go. So this is your double helical structure of your DNA. So that is why it's called as, that's why it is said the nucleic acids have a lot of secondary structure. So this is your double helical structure. Right. So this particular turn is 36 degrees and you have 10 base pairs in this turn. And the height of this turn is your 34 Armstrong. So here I have one base, say for example A, so it will pair with T. If I have G, it will pair with C. But between A and T there are 2 hydrogen bonds, and between G and C there are 3 hydrogen bonds. But the distance between these two is 0.34 Armstrong or 0.34 nanometer. So this is the structure of your DNA, which I want you to remember here as well. Alright, so now coming and talking about the concept of metabolism. So what is metabolism? So metabolism is happening in your body as well as in my body, right? So you agree? Any change in that? Alright, so right now if you have had lunch, so if you have had chai, so your body is doing the digestion, right? So any major physiological process is your metabolism. Okay, so here we have certain concepts regarding metabolism. So biomolecules present in certain concentrations, they are expressed as molecules or moles per cell or your moles per liter. So that is how you express something. So if you ask me how many likes I have got I give you a qualitative number, right? So everything has an amount of proportion. So how much of salt did you add in your food? So that has a value, that has a proportion. So similarly, all biomolecules, the carbon containing compound in your living tissue, it has certain value, so it has a certain quantity. So it is expressed as moles per cell or moles per liter. So biomolecules have a continuous turnover. For example, by the process of digestion, if the 𝐕𝐴 is broken down into glucose, this glucose will be used for some other process, right? So it is not present in one form always, it is continuously changed from one form to the other form. Right? So that is my turnover. So always glucose does not remain as glucose. So probably it can be stored as glycogen. It can enter your Krebs cycle. It can go on to a link reaction. So a lot can happen, right? So that is your metabolism. So each metabolic reaction results in the transformation of your biomolecules. For example, even the removal of carbon dioxide from amino acid is your metabolism. So removal of amino group in a nucleotide base, hydrolysis of glycosidic bonds in a disaccharide. So everything is an example of metabolism. So metabolites are converted into each other in a series of linked reactions, that is the metabolic reactions. It is a continuous pathway. So that is how it has to flow. So it is already being defined in your body. We are trying to study that, right? So there's a particular direction in which your metabolites flow. The product of metabolism is your metabolites, and there's a particular direction in which it flows. So the metabolite flow is the dynamic state of body constituents. So the rate at which your metabolites are being produced, that defines how dynamic you are. So that you would have always had a friend in your group who eats so much but still does not put on weight. So probably the metabolism is good, right? But every metabolic reaction is a catalyzed reaction. So you have a catalyzer, you have an enhancer. We have already read about okay, so this is the first time we are reading about biomolecules, alright. So we have read about enzymes, right? So they are biological catalysts. Even for water, carbon dioxide dissolving in water, so that is also catalyzed by your enzymes. So catalysts actually hasten the rate of metabolic conversion of proteins. So basically enzymes are your biological catalysts. So this is how your metabolic pathway is interlinked. Alright. Okay. So metabolic pathways can be a more complex structure from a simple structure. So metabolism can be amphibolic, I would say, just like your Krebs cycle. So it can be more catabolic as well as anabolic. So catabolic is breaking down larger molecules into smaller ones. Anabolic is building of larger molecules from the smaller ones. But your metabolic process includes both of them. Alright. For example, acetic acid becomes cholesterol, right? And this is your catabolic pathway, right? So acetic acid breaks down to become your cholesterol. For example, glucose becomes lactic acid in a skeletal muscle. So this is your anabolic reaction. So anabolic reaction actually consumes energy. So anabolic reaction is like climbing up the stairs, which requires energy. Catabolic reaction is climbing down the stairs, which does not require energy. So in your catabolic reaction, so when you're climbing down, energy is released, right? So in catabolic reactions, energy is released. In anabolic reactions, energies are utilized, okay? So the energy that comes out during the catabolic reactions have to be captured and stored and trapped so that we can use it for the anabolic reactions. So where can we store this energy? So here comes the high energy molecules like your ATP, where energy can be conserved. So living organisms have learned to trap the energy liberated during degradation and store it in the form of chemical bonds. As and when needed, this bond energy is utilized for biosynthetic, osmotic and the mechanical work that we perform. The most important form of energy currency in living organism is stored in high energy bonds, that is your adenosine triphosphate, that is your ATP. So I hope you guys have understood, right? So before we go to the questions, so your metabolism is of two types. One is catabolic, the other one is anabolic, right? So catabolic means where bigger molecules are broken down to form smaller molecules. So in this process, energy is released, right? So anabolism, so smaller molecules are joined to form bigger molecules for which we need energy. So we have to trap this energy so that we can use it in the anabolic reaction. It is trapped in the form of high energy bonds, that is your ATP. That is what is being explained in this slide and in the previous slide. Alright, so before we go into the topic of enzymes, which will be starting from the next class, so here we go and talk about certain questions that are pretty easy that comes from your biomolecules. So I guess I have 5 questions now. So identify the basic amino acid from the following. So when I was talking about amino acid, I told you amino acid can be classified into a variety of ways. So you have plethoric ways in which you can classify the amino acids, right? So essential, non-essential, aromatic, aliphatic, hydrophobic, hydrophilic, so on and so forth. So when I was talking about the polar amino acids, so you have positively charged, negatively charged, right? So among them, the positively charged are the basic amino acids. From the given option, can you tell me which one is a basic amino acid? It can definitely not be glutamic acid because it is acidic. So you have a doubt between tyrosine, lysine and valine. So what would be your guess? So your lysine is the basic amino acid. So option C would be the right answer. So uridine present only in RNA is a nucleoside, nucleotide, purine or a pyrimidine. So uracil is a nitrogenous base. So it's a type of pyrimidine. So this combines with the sugar, that is your ribose sugar, and it forms your uridine. So this is your sugar. So sugar and a nitrogenous base, it forms your nucleoside. So the nucleoside now combines a phosphate group to form your uridylic acid. So uridylic acid is my nucleotide. So uridine is my nucleoside. Alright. Okay. So question 3. Which of the following is the simplest amino acid, the most easiest amino acid? So I write the structure of amino acid. So you have a H group. So you have a carboxylic group and you have an amino group. So this is nothing but a R group. So instead of this, if I write H, that is the simplest amino acid. The simplest amino acid is your glycine. Alright. So which one of the following is a non-reducing carbohydrate, meaning it does not have a free aldehyde group? So that is your sucrose. So sucrose is a non-reducing aldehyde. Alright. So what is the composition of lactose? So lactose is not a polysaccharide, but lactose is a disaccharide. So lactose is made up of glucose plus galactose. So this is the composition of your lactose. Alright. So I guess we are done with all the 5 questions and the chapter will be continued. So we'll be continuing with the concept of enzymes. So how enzymes work, how enzyme enables a reaction to form substrate. So what are the different properties or features of an enzyme that you should consider? So what are the factors that affect an enzyme? So what is the classification? What is the nomenclature of enzymes? So all this we'll be talking very soon. So until then, so we'll stop here. So Friday we do not have a class because it's Holi. So wishing you all a very happy and a colorful and a prosperous Holi. May the beauty of colors bring a lot of joy and prosperity in your life. So wishing you and your family a very, very happy Holi. So I'll meet you next week. So we'll finish off with the concept of enzymes. So until then, this is me signing off. So take care and have a great weekend ahead. So I'll meet you very soon. So thank you and bye.